instructor of BBT315 Metabolism, and today we will study lecture 2. This is the overview of metabolism. Okay, so all the product of digestion are metabolized to acetyl-CoA oxidized by TCA cycle. So breakdown of food stuff here. Carbohydrate will produce glucose, and glycogen, that is a major food, will also that is a major source of stored carbohydrate in the liver, will produce glucose. Protein will produce amino acid, triacylglycerol will produce fatty acid. So this glucose, after glycolysis, will produce pyruvate. This pyruvate will produce acetyl-CoA. And amino acid can either directly be produced acetyl-CoA, or can be converted pyruvate, then pyruvate to acetyl-CoA. And fatty acid directly produce acetyl-CoA. So this acetyl-CoA is a major fuel that can enter into the TCA cycle to produce carbon dioxide and water and energy. And sometimes TCA cycle involved in the production of ketone bodies. Okay. So what about the carbohydrate metabolism? If you summarize the carbohydrate metabolism, it looks like same here. So glucose, the major fuel of most tissues, and metabolized to pyruvate aerobically and lactate anaerobically by the pathways of glycolysis, which further gets reduced to acetyl-CoA. That means when you take glucose in the diet, it will produce glucose phosphate, triose phosphate. That means glucose will first convert it to pyruvate through glycolytic pathway, and then if there is anaerobic respiration, the pyruvate is converted to lactate, or if there is aerobic respiration, the pyruvate will convert it to acetyl-CoA. This acetyl-CoA enters into the TCA cycle and producing energies and carbon dioxide. And sometimes the amino acid that is released from the TCA cycle will produce protein, or amino acid can be produced from pyruvate also. And sometimes acetyl-CoA can contribute to the production of fatty acid, or cholesterol, or triacylglycerol, or sometimes the triose phosphate that has been generated from glycolytic pathway can produce DNA RNA. Okay, that means the pentose sugar of DNA RNA. And sometimes glucose 6-phosphate can enter into the pentose phosphate pathway to keep your body free from oxidative stress. Okay, so let's have an overview of metabolism. And here the carbohydrate can be converted to acetyl-CoA, and amino acid also can be converted to acetyl-CoA. And then this acetyl-CoA can be converted to fatty acid through lipogenesis, and fatty acid can be converted to triacylglycerol through esterification. So from that, basically we intake fatty acid. That fatty acid can goes through beta oxidation to produce acetyl-CoA. Then this acetyl-CoA enters into the TCA cycle. So I repeat, in the diet we take lipid. This lipid is converted to fatty acid. This fatty acid will convert it to acetyl-CoA through beta oxidation, and then this acetyl-CoA will enter into the citric acid cycle, and through the cyclic acid cycle it will produce carbon dioxide and energy. Okay, and sometimes the acetyl-CoA can also contribute to the production of cholesterol and finally steroid. So source of long chain fatty acid, either dietary lipid or de novo synthesis from acetyl-CoA. And fatty acid may either get oxidized to acetyl-CoA through beta oxidation, or esterified with glycerol forming triacylglycerol, the body's main fuel reserve. Okay, so let's check the amino acid metabolism. Here we take amino acid from the diet. So dietary protein converted to amino acid. This amino acid can contribute to the production of tissue protein, or this amino acid can contribute to the production of non-protein nitrogenous derivatives, or tissue protein can also give rise to amino acid. This amino acid can be can go through transamination reaction, and where they can convert it to carbohydrate, they can be converted to ketone bodies, they can be converted to acetyl-CoA, they can be converted to nitrogen, amino nitrogen in glutamate. So this amino nitrogen can be released from amino acid by deamination reaction and contribute to the production of urea. And the acetyl-CoA that has been produced from amino acid will enter into the TCA cycle to produce carbon dioxide. Okay, so the diversity of metabolism pathways in plant and bacteria. So pathways are photosynthesis, nitrogen fixation, oxidation or reduction of inorganic minerals, acid and gas producing fermentation. And what are the organism like plants and cyanobacteria? Photosynthesis occurs in plant and cyanobacteria. Nitrogen fixation occurs in specialized salt bacteria. Oxidation and reduction of inorganic minerals will occurs in archaeobacteria, and acid and gas producing fermentations in anaerobic bacteria. Metabolism of carbohydrate. So what about carbohydrate anabolism? That means synthesis of carbohydrate. Plants synthesize their own carbohydrate via photosynthesis from carbon dioxide, sunlight, and water. And in case of animal and fungus, there are two processes of production of carbohydrate. One is gluconeogenesis and another is glycogenesis. So gluconeogenesis is a method of carbohydrate production from non-carbohydrate sources. It occurs in animal and mammals. The site of gluconeogenesis is liver in vertebrates. There another method of carbohydrate synthesis, glycogenesis, the process of conversion of glucose to glycogen. And carbohydrate catabolism, the breakdown of carbohydrate like glucose happens via glycolysis. Glycolysis happens in both plants and animal, and the breakdown of carbohydrate like glycogen happens via glycogenolysis, a catabolic reaction process. So vital role performed by carbohydrate are those are storage of energy like starch and glycogen, structural component, cellulose and chitin. Carbohydrate works as a coenzyme component like ribose of ATP, FAD, NAD. It works as a component of nucleotide like deoxyribose of DNA, and it has role in the immune system and fertilization. It also has role in growth and development. So what is the overview of metabolism of protein? What are amino acids and proteins? So we know amino acids and proteins are the basic structural unit of all the cell. Proteins are building blocks of many biological entity, and proteins are actually polymers that are made of monomers called amino acids. So amino acids are organic compound having two essential groups, amino group and carboxyl group. And then there is one side chain group that is specific to each amino acid, and different or same amino acids are linked to each other by peptide bonds and form long peptide chain. So anabolism of protein. Protein anabolism is the process by which various proteins are synthesized inside or outside a biological body. It encompasses two processes, amino acid synthesis and protein synthesis or polypeptide synthesis. Amino acids are two types, essential and non-essential in humans. Non-essential ones can be synthesized by human body, but essential ones need to be taken via diet. There is no such concept of essential and non-essential amino acid for plants since they can produce all of them. Protein synthesis from amino acid encompasses four major steps: transcription, translation, post-translation modification, and protein folding. So what about the protein catabolism? The breakdown of protein is specifically called proteolysis, and this is the protein catabolism. After the protein are breakdown to the monomeric unit like amino acid, they are further reduced by degradation into individual atoms like carbon, oxygen, nitrogen, hydrogen. Okay, and the vital role performed by proteins are catalysis of metabolic pathway as an enzyme, formation of cell and organism structure, intercellular transportations, cellular signaling, antibodies and immune functions, hormones and storage. So metabolism of lipid synthesis. Lipid anabolism is the process by which lipids are synthesized biologically, and plants are the major procedures of lipid on the planet. Plants and bacterial systems are characteristically different from animals and mammals in the lipid production since they processes distinct enzyme for each step of lipid biogenesis, while the animals and mammals process a multifunctional enzyme that carries out all the steps of lipid synthesis. Plant processes a special desaturase enzyme that aid in the introduction of double bonds after carbon 9 and 10. Mammals are devoid of any such enzyme and thus cannot synthesize their own omega-3 fatty acid like omega-3, omega-6, and making these are essential fatty acids that need to be obtained from the diet. Okay, so the catabolism of lipids is the process of lipid breakdown through beta oxidation. Site of beta oxidation in plants peroxisomes and glyoxysomes. Site of beta oxidation in animals and mammals are mitochondria and peroxisome. Glyoxysome is absent in mammals. So, what is the vital role performed by lipids? This storage of energy and structural component. Lipids are the storage form of energy. Lipids are the structural component of membranes. Hormonal and homeostatic functions. They are signaling and chemical messengers, and their work is a cushion for vital organs and transport fat soluble nutrient. So let's study the metabolism of nucleic acid. So nucleotides, those are the basic building structure and functional block of nucleic acid. And nucleic acid anabolism. To study nucleic acid anabolism, we know all living cells are capable of producing nucleic acid, and dietary intake of nucleic acid isn't really needed. Nucleic acid from the diet are actually degraded and reverted back from some more anabolic type metabolic reactions by salvage pathway, anabolic type metabolic reaction. So we'll study that one later. And catabolism of nucleic acid or nucleotides is a continuous process that keeps going on as the nucleotide anabolism. There are only two possible end destination of nucleotide. One is degradation into waste product and final excretion, and second is the re-entry of catabolized products via salvage pathway into nucleotide production. So here is the flowchart. Catabolism: DNA to RNA, nucleotides, nucleoside, bases, uric acid. And formation, anabolism or salvage pathways, bases will reverse back to produce nucleoside, nucleotide, and DNA. So the vital roles performed by nucleotides are: these are storage in storage and transfer of genetic information. They are co-substrate and coenzyme. They're involved in cellular signaling. They're source of phosphate group.